I think I'm just gonna get started on the introduction while we're still waiting for a few people. So good morning, everyone. I'm very excited to see all of you here with us today. My name is Wasmon, a science specialist at GFI APAC. I would just like to give a short introduction about GFI for those of you who may not be familiar with GFI ecosystem. GFI, or the Good Food Institute, is a non-profit organization focused on advancing alternative proteins, which we define as plant-based, fermentation-derived, or cultivated meat, egg, and dairy products. We are proud to be 100% funded by philanthropy, which allow us to stay objective to our mission. Before we start out our seminar today, just a few logistic points. For the Zoom webinar, if you look at the bottom of the screen, you can see the Q&A button next to the chat button. Just want to highlight that if you have any question during the seminar, make sure that you put that into the Q&A button. And for any other comments or saying hi, where you come from, you can put that in the chat box. With the logistic aside, let's move on to our seminar for today. Today, I'm very honored to be joined by Dr. Raffael Osen. Dr. Raffael is currently leading the food process engineering group at SIFBI A-Star in Singapore. He's particularly interested in integrating protein ingredients development, texturization, and formulation from a wide range of alternative protein sources, such as underutilized Asian crops, fermentation-derived proteins, and cultivated cells for developing tasty, healthy, and sustainable and affordable foods. Dr. Osen started his research on development of plant-based meat substitute in 2010, while he's obtained his PhD in food technology from Technical University of Munich on high moisture extrusion of plant protein. Prior to his move to Singapore, he led the Department of Food Process Development at Fraunhofer, the largest organization for applied R&D in Europe. In his 10 years of experience, Dr. Osen has been awarded for numerous competitive research grants and industrial funds in the field of protein extrusion. He authored 15 peer-reviewed research papers, a book chapter, 3 patent applications, and was invited to give keynote lecture in several international food tech conferences. Therefore, I'm very excited to have Raffael with us today, and I'm sure we'll learn a lot from him. With that, Raffael, the stage is all yours.
All right, thank you, Wassermann, for this very kind introduction. I think I'm blushing now. I'm not sure if you can see that. Fantastic to get such a run through by you. Yeah, thanks very much. It's a great honor really to be part of this seminar and actually to kick it off, the GFI APAC seminar series, The Science of All Proteins. So I've been a big fan of the, let's say, the US version or the international version of that seminar. I've been following this since many years, and great to be able to be part of this. So I want to start by talking a bit about what is happening right now in Asia in the alternative protein space, and specifically what is happening in Singapore, and why I think Singapore is such an exciting place to be right now in the area of alternative protein and all these kinds of developments. Now, if we look at this graph here, it shows the map of Singapore. It's a rather small country, about 5 million plus population, but I think it's pretty impressive to see the amount of companies that are coming into Singapore to do research and to do developments in the area of not only alternative proteins, but food and beverages in general. But we do see also several companies, ingredients companies, for example, and food producers that are anchoring in Singapore, and from there going into the Asian market. So there is a lot of attention and a lot of traction right now from the private sector. Another I think important aspect is the legislative framework that Singapore has set, and I think it is already well known that the first cultivated chicken is available in Singapore, and I just heard that now it's even available in the hawker food stalls, which is the Singapore version of Asian street food, which I think is very fantastic. and the Impossible Burger has been improved. So I think Singapore shows really, it sets a good example of how, how changes can be made in order to really push for new developments in the quest for making the food production system more sustainable, more nutritious, and more delicious. So we have the private sector, we have the legislative framework, and then with this graph here, I want to show also the strong support by the government. And this is an example of a framework called the Singapore Food Story, which is about 1 million dedicated funding pot for research in agri-food, in order to support the very ambitious 30 by 30 goals that the government has set, which means to increase the food production up to 30% by the year 2030. And we can see here that in this, let's say, in this square here, that one specific focus is on the topics that Wassermann mentioned already that are important for GFI, which is the cell-based meat, microbial protein, as well as plant proteins, and then developing tasty and delicious and sustainable foods out of this. So I think this is also a very good, a very good sign for everyone who is into research and who wants to who wants to get some good funding, come to Singapore and then do the research with us. In order to do that research, there is a large ecosystem in Singapore based encompassing different IHLs, institutes, universities that are spread all over the country. But spread all over the country does not mean you have to travel far. Actually, everything is probably within 1 hour range, so it's very convenient to do project meetings and to collaborate. Now, I want to give a quick intro of A*STAR, being the agency for science and technology research in Singapore. It's a government agency under the Ministry of Trade and Industry with about 18 research institutes, about more than 4,000 people staff, a very international group of scientists. And the mission of A*STAR to drive applied research, translational research that in the end leads to consumer products that can be then enjoyed by everyone and by us being also consumers ourselves. And I think this is also a great motivation to do the research in such an agency or in such an institute. This graph shows an overview of the different of the wild field of technology areas that A*STAR is active in, and with a special notice here of one of the most recent institutes, which is the Singapore Institute of Food and Biotechnology Innovation, which I'm today proud to represent. At the Singapore Institute of Food and Biotech Innovation, SIFBI for short, we have basically we are structured into 5 departments where we can then look at the whole value chain of food production, and again, a special emphasis on alternative proteins. This comes from more the biotechnology angle, and here we have colleagues that are in our discovery platform that have a large a physical library of more than 5,000 edible specimens, which is one of the largest libraries in the world, where they can then mine for new molecules that can be ingredients such as flavors, such as colorants, or new protein sources, which then are taken over by our colleagues in the strain engineering department, who then take these molecules, these identified molecules, and then use yeast or use microorganisms to then express these kind of high value ingredients, which then go over to our biotransformation colleagues, who are experts in the scale up, in upstream and downstream fermentation, not only on high value ingredients, but also biomass from fungi, for example, or microbial biomass. So here we really cover then the whole range of the fermentation side. We also have colleagues who are working in cultivating cells. And then all of these ingredients are taken over then to my team, the food process engineering team. We are about a group of 20 people. And then we look on the one side into how can we texturize these kind of new, novel, alternative protein ingredients. We also do research in processing or in fractionation of underutilized Asian crops or agri-food side streams into ingredients, so with this we can really cover the whole range of protein ingredients and then structure those and then hand these over to our nutrition colleagues who then look into what is the effect of these ingredients or of these food products that we develop on the metabolism, what is the metabolic response, with specific consideration of the Asian phenotype and how the different someone from China or from India or from Malaysia, how these groups react to these specific foods in terms of nutrition and also in terms of sensory properties. And with this, let's say, quick introduction, I would like to start with the topics of extrusion texturization of plant proteins. Now, the unique, let's say, situation and the opportunities of doing this kind of research in Asia are based on the fact that actually traditional Asian mock meat is not something new. In Europe and the US, this idea of having meat substitutes is a rather new concept coming from the last maybe 10 to 15 years, whereas for Asian consumers, this has been rooted in their diets. So the opportunity here is to increase the nutritional value and the sensory value of these kind of mock meat products that are used in the diets, but that are in some cases seen as probably not as high quality as the real meat, right? So this is one opportunity. Another one is to adapt the meat substitutes that are coming in now from more, let's say, the Western countries. And here's a burger as an example. How can these products that are more realistic, that are more similar to real meat, how can they be adapted to match the sensory preference as well as the nutritional preference of the local consumers? And then another opportunity is to use underutilized Asian crops as protein sources or agri-food side streams. So in order to do that, it's important to, on the one side, understand the production process of the protein ingredients as the bulk ingredient for these kind of products, as well as then the texturization process. And the probably most used texturization process until now is extrusion. And this graph shows the low moisture extrusion cooking process, which has been developed since the 1960s, which basically uses a defatted soy flour or a protein concentrate, which is mixed with water to a water content of 20 to 40%, which is then pushed through either one single screw or a twin screw extruder. It is heated, and temperature, shear, and pressure then result in physical chemical reactions of the mass, which then, after pushing the mass through a small orifice die, then the mass expands, the water evaporates, and after drying, then this leads to a foamed kind of, or a fibrous network that then after rehydration can be used in formulated products such as this burger that is shown here. Another version of this process is a high moisture extrusion process, which uses higher water contents, higher protein contents during the processing. And then instead of pushing this material through a small die, it is pushed through a long cooling die, which facilitates a laminar flow profile. It avoids the expansion of the material due to water evaporation, and then facilitate a texturization in a laminar flow profile, which is driven by protein cross-linking and phase separation phenomena. The products are shown here on the top right. It's a more of a gelled fibrous network with a high water content, 60 to 80%, and this can be used, for example, for muscle-like products such as strips, chunks, or steaks. Now, the one advantage of the extrusion process is that it's a multi-step process that can be scaled up and that can be industrially used. On the other side, this one-step process makes it also challenging in terms of product development, because there are many influencing factors that all are that all are intertwined. So whenever changing one process parameter, then the rest of the process parameters are changed as well. And in this graph, this basically breaks it down into 3 categories: the feed, the raw material categories on the top, and the machine let's say, parameters such as the screw profile, the extruded type, the size, the cooling die, and then the process parameters, the screw speed, mass flow, cooling temperature, and so on. Now, all of these independent process variables that can be adjusted, they affect the dependent system parameters during the extrusion, which then, such as residence time, pressure, viscosity, shear, and all of these have an impact on the physical chemical reactions that are happening as both in the extruder as well as the cooling die, which then affect the fiber formation. So this is to show really that, you know, changing one of these parameters really has an effect on the rest of the process, and therefore this creates, let's say, this leads to many times trial and error approaches during product development. Now I want to show some examples of how protein ingredients can affect the processing behavior and texture properties of high moisture meat substitutes. And the motivation for this work was really coming from the question, from the observation that if we have one raw material category, such as, let's say, soy protein concentrate or P, and we have different suppliers, then many times the extrusion behavior is very different, the texture properties are very different, and there's not that much literature available on these effects. So when we started this work, most of the literature was done on soy protein plus wheat gluten. So then we used a P as an alternative raw material, and then looked into how can we come up with a process, high moisture extrusion process, to texturize pea protein, having a simple model recipe, and for that we use pea protein plus water. And then how can we, by characterizing the protein, can we find out some explanations of why the proteins might have different extrusion behavior or different extrusion texture properties. So the method to test for the texture properties is very simple. It's a cutting strength, testing the cutting strength using a texture analyzer in length and crosswise directions, whereas similar values are indicated for a uniform structure, and different texture values in length and crosswise directions are indicated for a fibrous structure. Now, looking at the graph below, this shows the extrusion or the extrudate shape after increasing the extrusion temperature. And we can see that with increasing the temperature, then starting from 100 degrees C, there is more a dough-like texture, and with increasing the temperature, some fiber structures are formed. Now, looking at the texture properties, this can also be seen in the graphs here, showing the cutting strength and the cooking temperature, that with increasing cooking temperature, the lengthwise fiber strength increases. We do see a small, let's say, difference in protein ingredient number 2. And this is shown here at the temperature below 130 degrees C, with some variation in the texture strength. And this was shown also in looking at the extrudates, showing a more ununiform fiber formation or ununiform structure. So it seems like that the temperature was not enough. The thermal energy was not enough really to texturize these proteins into a fibrous texture. And this can also be seen here on the graph on the right, which shows the specific mechanical energy of the different ingredients. And we can see that the highest energy to process the materials was required for protein number 2. So looking at the texture properties, we do see some differences between 3 protein isolates, pea protein isolates. In this graph here on the left below, it shows again the specific mechanical energy input, but here it shows we're showing the startup phase, meaning that the time after the extruder had started, we record the SME for 3 different protein isolates, and we increase the temperature from 40 to 100 degrees C. But what can be seen here again is a significant difference in the processing behavior, showing a very low initial specific mechanical energy intake during the processing. So thinking back of the graph before, where we saw that at extrusion, at high extrusion temperatures, protein number 2 required the highest mechanical energy. Here we see the lowest mechanical energy. So obviously there's something happening with the proteins during the heating process. And our first assumption was, well, maybe it's the particle size, that the particles of protein number 2 are just so large that it takes some time for them to swell, to take up the water and to really form a dough. And therefore we might see here this curve progression. Now, interestingly, looking at the particle size, we found that protein number 2 actually is right just between the other two protein ingredients. So most likely this cannot really be the explanation for this behavior. We then looked into the composition as well as the protein, the amino acid profile to find some answers to that. And looking at, starting with the composition here on the top, we can see that it's rather similar for all three protein ingredients, with a slightly higher protein value for protein number 2, which would not really explain why we see this kind of difference in the behavior, in the extrusion behavior for that ingredient. We then looked into the amino acid profile, thinking maybe it's a difference in the cysteine. Cysteine can form disulfide bonds, which is important for the texturization process. Here we also didn't find any hints to explain that kind of different behavior. We looked into the molecular weight distribution, and also here we can see that the banding pattern is fairly similar between the protein number 1, number 2, number 3. In non-reducing as well as reducing conditions, we can see the legumin bands that are then dissociated into legumin alpha and legumin beta. Under non-reducing conditions, there's viscillin, there's conviscillin. However, what we do see here is a slightly more intense bands of legumin for protein number 2. So this could be indicated for a higher protein solubility. So we looked into that, and we did see that in the low and high pH range, that protein number 2 did have a higher protein solubility compared to the other protein ingredients, which would explain partly this difference here in the molecular weight intensity. Then next we then looked into the denaturation, thinking of, okay, if the solubility is higher, this could be indicative of a not fully denatured protein, and the data here shown the differential scanning calorimetry graphs basically reassured us in that thought that protein number 2, and we found a endothermic peak. In comparison, protein number 1 and number 3 did not show any of these endothermic peaks. So this shows that protein number 2, as a result of the processing history, was not fully denatured and therefore had a higher protein solubility. And this can be explained by the folding of the protein, the structure of the protein. And usually for native proteins, the hydrophobic side chains, they're more trapped inside the molecule. When during the denaturation process, then the structure changes and partly unfolds, and then the hydrophobic side chains, they become available on the surface. And the same thing can be seen here for the surface hydrophobicity, that for protein number 2, we do see the lowest surface hydrophobicity, which correlates very well with the denaturation. And then as a result, looking at the technical functional properties, we can also see these effects for the water binding capacity, oil binding capacity, as well as emulsifying capacity, where then protein number 2 has significantly lower values compared to the other proteins. Now coming back again to the extrusion behavior, this is really, or this can be a critical part during the startup phase. Everyone who's working with extrusion probably has experienced that themselves. And we then compared, or we thought, okay, can we maybe have some simple rheological tests in order to anticipate how a protein could react or could behave during the extrusion. And if you look at this very simple RBA setting where we compared in protein number 1, 2, and 3, and we look at the extrusion behavior during the startup phase, it's a fairly similar curve progression with a high swelling in the startup of this process, which can be attributed to the high denaturation degree and to the high water binding capacity of these ingredients in comparison to P protein number 2. So this means that from an, let's say, application point of view, this kind of very simple RBA test can help to get an idea of how a protein could behave during extrusion, and especially with very with a very high solubility, which is for many applications sought after. It can actually be a bit of an issue during the startup phase for extrusion. So this is an example to show that having that characterizing the protein ingredients to be used can help really in, on the one side, understanding why the differences are present during the extrusion process, as well as for the product quality, and then how could this be used in the future to anticipate how a protein might behave during extrusion. Now, in order to change in order to develop a certain structure of extrudates, a certain lengthwise or crosswise fiber structure that can be used then into different, let's say, meat, pork, chicken, or beef products, it's important to understand the rheology, the viscosity, the flow profile during the cooling die. And this is all, again, is affected by the protein structure. So one way to to understand better of what is going on during the extrusion and during the cooling die passage would be to do rheological tests. Now, the challenge here is that the materials, and this can be seen here in the graph below, usually they form a very crumbly dough-like structure at a protein concentration that is similar to the protein concentration that we have during extrusion. So it's difficult to do a viscosity test. Now, based on the knowledge or based on the findings that I presented before, that P protein number 2 was less denatured and therefore had a higher solubility. And this allowed us to do some tests here in a pressure cell rheometer, where we then were able to have similar protein concentration up to 60%, while still have a homogeneous dispersion that allowed us to do some rheological testing to understand better the viscosity during the extrusion as well as during the cooling die. So we can see here the elastic modulus as well as then the viscous modulus and the changes during heating the material up to 150 degrees C. And we can see that how the materials, they start to form a plasticized mass in the cooling die, in the cooking zone of the extruder, and how the material then starts to harden and solidify again in the cooling die channel. And these results, they were the basis then for some work that we did towards simulation and modeling the processes during the high moisture extrusion process. So in summary, what I try to illustrate here is that the characterization of protein ingredients can help on the one side to understand differences in the processing behavior and in the product quality, and also to anticipate extrusion behavior and monitor the protein quality in terms of, for standard recipe development, for example, which is very important for the industry. It can also help to better understand the effect of protein fractionation on extrusion behavior and product quality. This is something that I have not shown here. This is something that we are working on currently, and that I think is important really to, at one point, be able to develop protein ingredients that are specifically designed for specific extrusion applications. I've been talking a lot about protein structure and about the extrusion process, the effect of the extrusion process. What I did not mention is that we also saw differences in the sensory properties of these ingredients. And here I want to give an example of some work that we did on how the processing can affect the sensory quality in protein ingredients and in food formulations and in the end. So our hypothesis was, and again, this example here is pea proteins, and everyone who is working with pea protein has probably also observed a certain beanie off flavor and a certain bitter astringent taste. It is reported that, or the hypothesis of this work was that saponins, amino acids, and peptides from hydrolysis, as well as lipid oxidation products, are one driving factor for these off flavors. So the approach was here to identify the key components that contribute to the bitter off taste and reduce bitterness during the processing. We started working with characterizing commercially available pea protein isolates, and then in order to understand better the upstream work, with the upstream, let's say, effects, we looked then into the processing of peas to pea protein ingredients in lab scale as well as in pilot scale. And this here is the process that is shown, which is a typical industry standard process where peas are dehulled, then they are milled into a pea protein flour, followed by alkaline extraction, centrifugation, isoelectric precipitation to a protein curd that is then washed, neutralized, dried into a pea protein isolate. This work was done together with our colleagues from the Technical University Munich, who did then the sensory characterization. And in a nutshell, what they found is that, yes, they basically they did find saponins and lipid oxidation products that had a very high intensity of these bitter and astringent tastes, but they also did find some new components that were found in the wax from pea hulls, which had a very a large contribution to this off flavors. And so that was surprising, and that was something new. Then the question was, how can we change the processing, the fractionation, the drying, the precipitation in order to improve the quality of the sensory quality of these ingredients? So one way to do that is to do a full dehulling, to really remove all the waxy components, to reduce that part that attributes then to the bitterness of the final ingredient. We also had the assumption that, well, if the lipid oxidation is a problem, how about we do a full de-oiling of the materials, and therefore, reduce that kind of problem. But in the end, it was we found that the de-oiling actually resulted in increased bitterness, which is most likely due to masking effect of lipids and sugars. So this was a new finding. This was quite interesting. Hydrothermal pretreatment also was shown to be effective in reducing the bitterness and reduce the enzyme activity during the processing. So there's still ongoing work on the influence of other conditions during this processing. So what this shows here is that the example here is on the effect of the processing on the aroma and on the taste. But obviously there are also effects on the functionality, and all of these, they need to be taken into account when going into the further processing, the texturization, and then the food application. So I talked about aroma. Now I'm going to give an example of how the processing can affect protein structure and the protein functionality. In this example, we have been using lupins. Lupin is a great source of protein, up to 40%, which grows very well in a moderate climate, such in Europe or in Australia, for example. So the idea was then, can we use basically lupin as an alternative to soy and develop some protein isolates based on lupin. The process was based on a industrially typical soy processing process, starting with dehulling and sifting, and the hulled flakes are then, the hulled seeds are then flaked to increase the surface area for the subsequent de-oiling. And then after de-oiling, the so-called white flakes are then treated by acidic aqueous extraction to remove the sugars of soluble plant secondary plant metabolites, and the residues are then treated by alkaline aqueous extraction to receive a protein extract where the fibers are removed, and the protein is then, after acidic precipitation, spray dried into a protein isolate. The quality of this protein isolate is made it possible to use that in a wide range of products. So this is some examples from the spin-off company Prolupine that was found. And besides these kind of products, also high moisture meat substitutes are possible to develop from these kind of protein ingredients. We also changed the processing parameters, especially in this case, the extraction and then the precipitation methods. And we found that by these changes, we were able to get a very different functional ingredients with very different functional properties. And this is shown here on the image below, which is a protein, a rehydrated protein that, which can be seen here on this image, almost has a fat-like behavior. And the reason for that is, if we look at the micrographs of this specific protein, we can see that it has a sort of micellular structure that is in the same size range as fat globules in comparison to the isoelectric precipitated protein. So small changes in the fractionation process can lead to very different functionalities that can be then used in different product applications. We also looked at the acidic aqueous extraction step and looked into the recovery of these proteins from the motivation of a sustainability approach. And when we characterized this protein that we got after ultrafiltration, then we found that it had a very unique and very interesting solubility curve. The yellow curve and the green curve, these are the isoelectric developed proteins that show this typically U-shaped curve with a low protein solubility at the pH range of 4.5, whereas then these the sour acidic soluble protein had a rather let's say pH independent solubility behavior, which then allows, for example, the application of these ingredients into sour or into acidic drinks, for example. Now coming back to the meat substitute and the extrusion texturization topic, what I try to show here is the innovation potential in using underutilized Asian protein crops and protein sources in the future, which makes it, you know, in order to do that, we really need to understand the protein functionality and the effect of the protein functionality on the final product application. And for that, we also need to understand the protein fractionation aspect. So in the end, we really need to look at the final product and understand what is the functionality of the specific protein, of the animal protein that is replaced by another protein, and then we need to work backwards by a reverse engineering approach and identify then the right processing chain in order to achieve this kind of functionality. We could see large effects of the protein fractionation parameters on the protein composition and protein functionality, and it is important in the future to move towards integrated processing for energy and cost reduction, and also to apply these methods then towards simulation and modulation and automation then for these texturization processes in the future. So this is my last slide, and here I just want to highlight again that I'm very excited to be in an institute where we have access to all sorts of alternative protein proteins coming from either microbial sources or coming from plant proteins or cultivated cells that we can then look into the texturization and then the nutritional effects of these foods. We do have the access, or we are working together with Temasek to build a food tech innovation center that will be launched soon. So we are very excited to do this work, not only in lab scale, but also translating that to pilot scale, which I think is also one very crucial aspect in terms of the industrial application. Then, so we are hiring. So anyone who is also excited about that, we are very happy to hear from you. And with that, I would like to thank everyone for the attention, and I'm looking forward to the discussion. Thank you.
Thank you so much, Raphael. That was really, really good. And the chat box has been exploding. This has been very active there. So just to let everybody know, because we got a few questions regarding the recording, this seminar will be recorded and published onto the Good Food Institute YouTube channel once it's available. So it's free access, so everyone can go check it out there. And for that, we're going to go into the question, but we have so many questions coming in, so we will try to keep the answer as short as possible to ensure we cover as many topics as time allows for today. So let's go on. The first question we have here is, in our laboratory, we're thinking of extruding plant protein as tissue culture scaffolds, but screw extruder are so expensive. Is there a lower cost lab scale method of extruding plant protein?
But there are different texturization methods available that do not really require having an extrusion setup. But from the question, it seems like it is about, is there another a cheaper extrusion lab setup? So I think if you want to have an extrusion lab setup, you would also need to get then an extrusion device. Otherwise, you might look into some simple shearing or some protein elongation methods that can be also used to develop some kind of an anisotropic structure. And I believe that is the goal here in this example, to have an anisotropic structure, a kind of fibrous structure, to have a scaffold where then the cells can then be attached and can grow on this network.
Thank you. For the next question, what is the length of cross-linking via extrusion, and how is the status of food safety aspect like GI fate of cross-linking via extrusion? Is there any clinical research for it?
Mm-hmm. Yeah, that's a very good question, and that is something that I see not really published a lot. There are some publications. I believe 2009, McDonald did some rat studies where they compare different soy protein, high moisture extruded soy protein isolates versus the non-extruded versions, and they found that there was no effect on the growth of the rats. We have been looking into the effect of the extrusion temperature, for example, on the amino acid composition, whether there is some reduction of certain amino acids, but we did not find any indications of that. But I believe that there is, there needs to be more research in that area.
The next question is, what are the pros and cons between hot and cold extrusion? Is cold extrusion commercially used in the industry?
Yeah, both processes are commercially used in the industry. I'm not sure what cold extrusion is referred to. So cold, I'm not familiar with a cold extrusion process for protein texturization. So if we talk about protein texturization, it would be low moisture and high moisture extrusion. So I'm not sure what exactly the question refers to. Cold extrusion is, again, for protein texturization. I'm not familiar with that. For the difference between low and high moisture extrusion, the pros and cons is that for high moisture extrusion, you get more of a fibrous meat-like, muscle meat-like texture, but it needs to be handled very carefully because it has a high protein content, high water content, so it's prone to spoilage. Whereas for low moisture extrusion, this is usually dehydrated, so it has a long storage time. It has, as an ingredient, it can be used, for example, for foam products, and then bring in this bite and and due to the foamy structure, the juiciness in a product, where, for example, high moisture extruded products, they might not bring in this juiciness. So it really depends on the final product application that you're looking into. And for some low moisture and for some high moisture extruded proteins, they work better.
Thank you. I think we need to go quickly here. Which structural and functional properties of alternative proteins are keys for meat alternatives, especially for high moisture extrusion applications?
Which functional properties, and what was the other part of the question?
Structural and functional property of alternative proteins are keys for meat alternatives, especially looking at the high moisture extrusion. Yeah.
Well, I would say the most important one is probably the taste, and is the color, and is the costs. So, and it's the safety, of course, right? Especially when we talk of alternative proteins that we might not be yet familiar with that much, that are in the exploratory stage. We need to make sure that all of these boxes are ticked. Then in terms of the functionality, like I have shown, if the particle size plays one role for the powder dosing, then for the extrusion itself, it is probably, it is probably the solubility that plays a role. And in this case, the solubility should not be too high, because otherwise you will get difficulties during the extrusion behavior.
Thank you. Next question, how do you know which proteins are legumins and mycelium? It is just from literature based on their molecular weights. Just curious if the molecular weights can vary for different protein sources. I think this referred to your, the protein gel, the SDS page.
Yes, right. So we compared the banding pattern to the literature, and then based on that, we did our assumptions on what are the changes in the protein banding pattern during the reducing and non-reducing conditions, which was very similar to what has been described in the literature. And that can be different, of course, for other protein sources such as lupins or lentils or chickpeas.
Thank you. The next question is regarding actually the shearing force inside the barrel. So can you please comment on how important are shearing force inside the barrel for fiber formation, or the fiber structure formation just take place in the cooling die?
So from our observation, the shearing in the extruder is probably not the most important parameter. It is mostly the residence time, as well as the temperature during the processing. Whereas the shearing with such high water contents, we didn't really see the large effects on that. The fiber formation mostly takes place in the cooling die during the solidification of the mass. Doing a dead stop experiment, you can also see some fiber formation already in the entry zone from the extruder to the cooling die. So there is a transition zone, which really depend again on the temperature profile that is used for the process.
Thank you. So the next question is regarding the protein fractionation process. Can you further explain why the de-oiling process resulted in an increased bitterness in the raw material?
Yeah, I mean, that is a finding that was surprising. And our idea was that probably by de-oiling, also some other components are reduced, such as sugars or salts, and these could have a masking effect of some bitter components. And by removing those, then in the end, the bitterness was perceived stronger than without de-oiling.
And I think this related to the previous question, like how does the oil content in raw material impact the fiber formation? Is it important on producing texturization product from oil
Yeah.
industry byproduct, like the oil press seed cake, I think.
Yeah, that's a very important question, and that is to our understanding very relevant. That the protein, the oil, is not so much about, in that case, for the fiber formation, about the effects of taste or aroma, but more on the viscosity during the extrusion process. That high amounts of oil are difficult or do prevent this fiber formation process, because it just reduces the viscosity, and it creates a kind of like a it reduces the shear forces during the process, so that the phase separation cannot really occur to such an extent that is required for the fiber formation. So therefore, this is a challenge. And especially when we look into agri-food side streams, such as, for example, oilseed press cakes, it really depends on the remaining oil content of these oilseed press cakes, and it might be necessary to also do a second de-oiling step in order to decrease it to such a level that then in the end this material can be processed accordingly.
Thank you. I have an interesting question here. As a manufacturer perspective, we are unable to get the deep information of raw material of protein from suppliers, such as protein molecular weight, viscosity, and so on. Should in-house lab work deeper on it for quality control?
If possible, I think this can help, and this does not probably require a highly sophisticated equipment that goes really into the molecular structures of the proteins, but maybe a more applied oriented method, such as the rapid visco analysis, for example, can help to do some standard screening of the materials that come in, and even with different batches, we have seen that from the same raw material source, from the same supplier, different batches, they showed different extrusion behavior and different texture properties. So doing a regular screening, for example, with rapid visco analysis or any other kind of rheological assessment, can help to get an idea of how this, whether there are any changes in the materials, or could also be used as a quality input control measure. So I think there's a lot of creativity in order to come up with very simple tests. It can even start by taking a powder and then just dispersing it in water, and then get an idea of the protein solubility, which then correlates with the viscosity to some degree.
Great answer. So the next question regarding the loop-in cell extraction. Does this process will impact on the sodium level in the final product? Does this mean this type of protein is soluble protein, and how can we apply into other applications?
That's a good question. I would need to look into that work. I'm not really, I don't remember exactly about the salt concentration of this. After the salt precipitation, I mean, what you can also do is then having a neutralization step afterwards, in order to take to reduce the salt content. But I would really need to look into that. So cannot really answer that clearly.
Thank you. And I got this question from Regini regarding the findings of differences in PPI2 with the others as a function of protein nativeness. Would one need to modify extrusion condition to achieve the same texturization as for PPI1 and PPI3? Thanks.
Yeah, that's right. That's what we assumed was the reason for let's say the higher energy requirement that was necessary in order to texturize P protein number 2. Compared to the other ingredients. So we had to really increase the extrusion temperature in order to achieve this texture formation that we have seen with the other ingredients. So yes, this is exactly right. You need, when you have different protein functional properties, you will have to change the extrusion process in order to achieve the similar results.
Thank you. Is there any standard pattern about thermorheological behavior of proteins to indicate the possibility in meat-like formation via high moisture extrusion processing?
Can you repeat again, Wasserman, please? Is there
I believe, is there any standard patterns about the thermorheological behavior of proteins? So I believe this regarding to like solubility, gelation ability, like protein functionality. Is there a pattern in your study? Is there a patterns from studying various protein that can kind of like confirm that it's possible to do extrusion, to do texturized protein via extrusion?
Okay, there are different rheological methods described in order to characterize proteins that have then also be extruded, and the relations have been described. So there is a lot of work from a fantastic group in Karlsruhe, and Dr. Azad Emin, who has done some work on closed cavity rheometry. And this describes the behavior at high temperatures, similar to the extrusion process. So a bit similar to what I have shown today on the high pressure cell studies, and that can help to anticipate and to better understand the polymerization process and the texturization process during the extrusion. I'm not sure if this answered the question, but that's my best take on it.
Thank you, Raffael. I think we have time for maybe 1 or 2 more questions, and I believe this one will be a good one to wrap us up. Could you speak on what technological bottlenecks, in your view, as a highest priority to tackle in plant-based food?
I would say from a consumer perspective, the bottleneck is still a limited range of raw materials that are available of protein sources. So I think here in order to reach more consumers, it is important to have a wider protein sources from also regional raw materials available. Another bottleneck is clearly the costs of the products. So it is important to work towards price parity, and for that, large volumes are required. So I think this is a bottleneck for developments of new protein ingredients that you need to start small, and then this will be much more expensive compared to soy, for example. And we saw that for the developments, for example, of the lupin protein isolate as an ingredient, that this can be a challenge. Another challenge is the sensory properties. That we need to understand better of how can we remove some of the bitter, astringent, or plant-associated off flavors. And then I believe it's also important or another bottleneck is that some ingredients that are used, they might be over-engineered for certain applications. A soy protein isolate, for example, probably for different applications, we might not need a soy protein isolate, and by that we could reduce the cost. So I think we need to have a wider variety, not only of protein sources, but also of protein ingredients in terms of the protein concentration. So concentrates, flours, as well as in isolates.
Thank you so much, Raffael. And we are running out of time. I really apologize for some of the answers we didn't get to address. So we will definitely keep this in mind when we do our next seminar. Again, thank you so much, Raffael, and thank you everyone for participating in this GFI APAC First SciTech seminar. Really glad to see all the attendance and see you here. And with that, I would like to close out this seminar. Thank you very much.
Thank you. Bye-bye.